Hello students, welcome to E-pgp Pathshala. I am Dr. Renu Tyagi from Department of Anthropology, University of Delhi. Today we will be discussing on the module Skeletal Analysis and Determination of Demographic Variables in Post-Neolithic Population Growth from the paper Human Origin and Evolution. The learning objectives of this module are: number 1, to learn the skeletal analysis, sex and age determination. Number 2 is to study the determination of demographic variable in post-Neolithic population growth. Now let's have an introduction about the topic. Population growth after the development of agriculture has been one of the most dramatic features of the human evolution, and the estimated population was 80 lakh individuals 10,000 years ago who inhabited the earth. By AD 1800, the world population reached 1 million. The traditional view given by the child holds that the post-Neolithic population growth resulted from the improved health, which may be attributed to the effect of the agricultural food surpluses. Explanation of the post-Neolithic population growth has focused on the change in demographic parameters. Most researchers agree that the population growth during the Paleolithic was very slow. Despite difficulty in estimating the Paleolithic and Neolithic population size, there have been a number of reasonable reconstructions. Lack of Paleolithic population growth has been explained by arguing that the population were experiencing maximum fertility and very high mortality. Neolithic population explosion resulted from the improved nutrition and health. These acted to reduce the mortality, and the change in demographic patterns led to a rapid increase in population. Sex and age determination through the skeletal analysis have been a very important source of determinants of demographic variables for the past population growth. Now let's have a look at the skeletal analysis. Male and female adult skeletal morphology varies across a continuum. Sex and age can be determined from the skeletal, which can be later helpful in assessing the number of individuals, age at death, fertility and mortality ratios, and population growth patterns. Now first have a look at the sex determination from the skull and pelvis. Much of the morphological differences between the sexes is genetically determined and is the result of two separate processes of natural selection. The sex determining genes control the development of the primary sex organs, which equip the individual for either the male or the female reproductive role. Because successful reproduction is the key to the survival of an individual's genes, these sex differences are under close control by stabilizing the selection. But the separate process of sexual selection also has an important influence on the visible or phenotypic differences between the gender. The bony pelvis, including the hip bones and the sacrum, is the most relative part of the adult skeleton for the sex estimation, because it is the only skeletal region which exhibits specific morphological adaptation to the different sexes' reproductive capacities. In the adult female pelvis, the postural and the locomotor functions of the pelvis are modified by the requirement for the birth canal to allow the passage of a fully developed neonate. This is achieved by additional transverse growth of the pelvic bone, which occurs around the time of puberty. This differential growth is manifest both in the overall shape of the pelvis and in a series of specific morphological traits, each of which contributed to the overall adaptation. After the pelvis, the skull, that is the cranium and the mandible, provides the next most reliable group of the sex-specific morphological indicators. The principal sex-determining features of the human skull are related to overgrowth or hypertrophy of the bone in the males, and to the tendency for males to show the greater development of the musculature and articulation with more prominent muscle marking and larger joint surfaces. For most of the sex diagnostic trait, of the cranium, the presence of the trait indicate a male and its absence indicate a female. Skeletal hypertrophy is seen most clearly in the skull of the adult male in the supraorbital region, the nuchal region, the mastoid process and the attachment areas for the muscles of mastication, and in the development of mental trigon, that is chin, on the mandible. When we move away from the pelvis and the skull, the sex discrimination relies principally on the average size differences between the sexes, particularly in the size of the articular surfaces at the end of the long bones. On average, the muscle bulk and the body weight of the male is considerably greater than that of the females, and muscle forces and static weight must be transmitted through the joint surfaces, which as a consequence are on average larger in males than in females. This figure shows the sex determination from the skull, and the next figure demonstrates the sex determination from the pelvis of the human. Now let's have a look at the age estimation from the ossification of the bones. The developing human passes through a succession of the well-defined stages. These are the embryonic stage from conception to the 8 weeks, the fetal stage from 9 to 40 weeks gestational age, the neonatal stage from birth to 1 month, infancy from 2 to 11 months, early childhood from 1 to 5 years, later childhood from 6 to 11 years, adolescence from 12 to 17 years, and adulthood from 18 years onwards. Bone and teeth begin forming in the fetus well before the birth. Ossification begins at the 8 weeks in the utero, that is in the womb of the mother, and the deciduous teeth begins calcifying at 15 weeks. Most of the bones of the skeletal are initially formed as a cartilage model, in the middle of which appears the primary center of the ossification. Secondary center of the ossification appears at the end of the bones and are separated from the primary center by intervening growth plate. These secondary centers subsequently become the bony epiphysis that supports the articular surfaces. Bone growth continues by the addition of a new bone on the surface facing the direction of the bone growth, while resorption takes place on the surface facing away from the growth direction. The crown of the deciduous and the permanent teeth develop within the jaw, and the teeth subsequently develop root as they erupt through the oral tissues into the mouth. Towards the end of the puberty, most of the bones stop growing and the cartilaginous growth plates underneath the epiphysis are converted to bones. The epiphyses fuse to their respective diaphyses at different times, but mainly from about 12 years onwards. Some growth centers in the skull and vertebral column fuse much earlier, while the medial end of the clavicle, the epiphysis of the vertebral bodies and the growth plates between the segment of the body of the sacrum do not fuse until the mid to late 20s. Most epiphyses fuse between 1 and 2 years earlier in the girls than in the boys, and fusion itself is a protracted process rather than a sudden event. This figure shows the age determination from the long bones including the humerus, radius, ulna, femur, tibia and fibula using their estimated age of fusion. Age determination of the fetuses need to be studied. During fetal and early neonatal life, the bones grow quickly with about 20 mm added to the length of the long bones diaphysis in just 10 weeks, compared to an average of 20 mm per year during the postnatal growth period. Thus, in principle, the age at death of a fetus can be estimated accurately from the measurement of the size of its bone. The deciduous teeth commence calcification between 13 and 20 weeks gestational age, but the crowns of these teeth do not complete formation until the postnatal period. The deciduous dental development proceeds faster in the prenatal males than in females. The tooth crowns are formed within the jaw and then erupt through the gum into the mouth, while the tooth roots grow downwards into the jaw. The stages of the tooth crown and the root development can be recognized either by the visual inspection or from a radiographs of the jaw with the developing teeth inside it. Up to 14 discrete stages of the tooth development can be recognized radiographically from the initial appearance of the calcification of the tooth cusps through to the closure of the root apex. Following the eruption, alternatively, the height of the crown of the developing tooth can be measured and compared to the standard growth curves calculated from the known age samples. The resorption of the root of the deciduous teeth prior to their exfoliation can also be used as a chronological marker in the children aged between about 5 and 12 years. As tooth development appears to be less influenced by dietary and other disturbances to growth than is the case for other tissues of the body, age estimates based on the dental growth are considered more accurate and reliable than those based on bone growth. An additional source of the data for the age estimation in children can be found in the dental microstructures. Teeth carry a record of their own development in the microscopic incremental structure in enamel, dentine and cementum. These structures are visible in the microscopic thin sections and electron microscope image of developing tooth surfaces. The incremental markers includes the striae of Retzia's weekly growth increments in the enamel, which are expressed on the outer surface of the tooth as pancomata and enamel prism cross striations, which represent daily variation in the enamel secretion rate. By counting the number of pericymata on the surface of a partially formed tooth and adding about 26 weeks to account for the hidden increment that are buried in the occlusal enamel, plus a correction for the age at which enamel calcification begins, the age at death of a juvenile skeleton can be calculated. This table shows the determination of age through the skull while studying the time of closure of different sutures according to Bardelle in 2011. The next figure displays the age determination from the skull with different sutures in the lateral, basal and top view of the skull. Now let's understand the determination of the demographic variable. Data from the ancient population, Europe and Asia shows that only a small proportion of the population lived beyond 50 years. Most died at about 60 or 70 years. In the case of the Middle East, Southern Levant, Herskowitz and Goffer studied the hundreds of skeletons that came from a hunter-gatherer series of the Neolithic period, that is 15,000 to 12,000 AP, and pre-ceramic sedentary groups from the Neolithic from 12,000 to 8350 AP. For the former, the estimated life expectancy at birth, assuming they are stationary population, is 24.6 years, while for the later group it is 25.4 years. The average age at death of the adult individuals represented in the series is 32.1 years for the hunter-gatherers, including 37.6 years for men and 30.1 years for the women, and it was 31.2 years for the pre-ceramic group, including 32.2 years for men and 35.5 years for women. In Roman Egypt, found that only about 1/5 of women lived from adolescence to age 60. The average life expectancy at birth was 25.6 and 22.5 years for men and women respectively. At 30 years, this indicator is 22.2 and 24.9 years respectively. Therefore, the probability of survival beyond the age of 50 was very low. From the study of the Chinese genealogies showed that very few people reached the age of 95 years and the life expectancy at age 30 and 50 were lower. Now let's see the mortality. It has been noted that age at death distribution constructed from the skeletal remains typically look significantly different than those produced by the historical demographer, even though the historical and the archaeological samples may not be that far removed in time. Paleodemographer, however, have typically reported unexpectedly low infant mortality and adult age specific mortality that is abnormally high and that accelerates rapidly from age 15 to 50 years, resulting in few individuals surviving into old age. Some anthropologists assert that mortality patterns in archaeological population were significantly different than those observable in the historic and the modern anthropological population. If skeletal elements are missing or incomplete, transition analysis allows for age estimation using only the features that are observable. One of the most significant benefits is the promise of improved estimates of age for older adults. Many methods of the adult age estimation have an open-ended terminal age category. While these open-ended categories acknowledge the difficulties of accurately aging older individuals, they also limit the information regarding the senescent mortality that can be obtained from the skeletal samples. Evidence suggests that the process of development since the Neolithic revolution was indeed accompanied by an increase in the extrinsic mortality risk. For example, skeletal remains from the pre-Columbian American analyzed by the Steckel demonstrate a decline in the health environment over the time period of 6000 BC until 1500 C. Importantly, difference in the timing of the Neolithic revolution across the region generated significant variation in the genetic composition of the contemporary human population. For example, lactose tolerance was developed among Europeans and the Near Easterners since the domestication of dairy animals in the course of the agricultural revolution, whereas in the region that were exposed to the dairy animals in the later stages, a larger proportion of the adult population suffer from lactose intolerance. The probability that an individual would survive to a reproductive age is affected positively by the genetically predetermined somatic investment and negatively by the extrinsic mortality risk that is associated with the socio-environment characteristics which were altered by the Neolithic revolution. A rise in the mortality risk triggers a process of natural selection that alters the distribution of type within the population. During the Neolithic revolution, life expectancy declined as long as the extrinsic mortality risk increased and the distribution of types in the population did not evolve considerably. However, the onset of the evolutionary process increased the prevalence of individuals with higher somatic investment in the population and ultimately generated a rise in the life expectancy. The transition from the Paleolithic period to the Mesolithic hunter-gatherer economies corresponded to the disappearance of the large game animals and the consequent adoption of the broad spectrum foraging patterns aimed at a wider array of small animals, seeds and aquatic food, resulting in a decline in the nutrition and thus human health and which lead to the mortality, according to the study done by Gellor and Mackay in 2007. Now let's have a look at the life expectancy. The rise in the population density, the domestication of the animals and the increase in the work effort in the course of the Neolithic revolution increased the exposure and the vulnerability of the humans to environmental hazards such as infectious diseases and led to the decline in the life expectancy during that period. However, that in the light of the fundamental trade-off between current and the future reproduction, the Neolithic transition altered the evolutionary optimal allocation of resources toward somatic investment, repairs and maintenance, for example, enhanced immune system, DNA repair, accurate gene regulation, tumor suppression and antioxidants. The rise in the extrinsic mortality risk, that is the risk associated with the external environmental factor as opposed to the internal biochemical decay in the course of the Neolithic revolution, generated an evolutionary advantage to individuals who were genetically predisposed towards the higher somatic investment, increasing their representation in the population and leading to the observed increase in the life expecting in the post-Neolithic period. The presumption predict that the interaction between the rise in the extrinsic mortality risk and the evolutionary process manifests itself in the observed non-monotonic time path of the life expectancy. In the short run, while the composition of the population remains rarely stationary, a rise in the mortality risk reduces life expectancy. However, the evolutionary process that is triggered by the environmental change generates an evolutionary advantage for individuals characterized by higher life expectancy, increasing their representation in the population. As the composition of the population shifts sufficiently in favor of the individual with higher life expectancy, the population's life expectancy increase and ultimately it could reach a higher level than the one existed prior to the increase in the mortality risk. This table shows the life expectancy from the Neolithic period to the Iron Age as given by the Aksadi and Nemskary in 1970. Now let's have a look at the population growth. The first period in the pre-modern time is Paleolithic period that lasts for 3 to 5 million years. Subsequent period, however, are indicated according to the scale, starting with the Neolithic period that concerned about 8000 BC and lasted until about 3000 BC. This was followed by the Bronze Age, which in turn were followed by the Middle Ages. The Middle Ages continued until the modern era that began around the 7th century AD. The growth of the population in the pre-modern times was very slow. Although reproduction was almost optimal in those times, the extremely difficult circumstances under which the people lived led to almost as much death as birth, with the result that population did not show significant growth. The slight growth that did occur was offset from the time to time by natural disaster, epidemics, famine and wars. Although a change from the nomadic to a more sedentary way of life in countries such as Mesopotamia, Egypt, Crete, India, China and Peru resulted in the establishment of well-ordered communities, it did not lead to a notable acceleration in the growth of the population. With the establishment of farming communities at the beginning of the Neolithic period, the world population started to grow slowly from an estimated 5 million to more or less 10 million around 8000 BC, more or less 300 million at the beginning of our era and more or less 500 million in 1650. Population growth has recently become a central issue in many archaeological models of culture change, yet only a few studies have addressed the problem of determining the population growth rates. Researchers provided an estimate of 0.1% for annual growth rate during the Neolithic on the basis of a guess estimate of the number of the inhabitants of Near East of the beginning of Neolithic, that is 50,000 to 1 lakh person, and about 1 to 12 million person at about 4000 BC. These figures indicate a probable rate of population growth between 0.08 and 0.12% per year. A lower rate of 0.03 was estimated for the period preceding the Neolithic and 0.24 for the period immediately following the Neolithic. The average for the Neolithic period was estimated at 0.1%. A report provided by an average of less than 0.1% from 1000 BC to about AD 1500, the average rate of population growth during the Paleolithic is exceeding in comparison with that of the Neolithic, and it has been found that until AD 1700, the human population grew at an average rate of 0.002% per year. The rise of the civilization seems to have been associated with the relatively rapid rate of population increase. This figure displays the world population growth from early agriculture to the modern era. Now let's have a look at the fertility. In demographic terminology, fertility refers to the number of children born to an individual, whereas fecundity refers to the physiological ability to bear the children. As with the mortality, natural pattern of the fertility characterizes human and animal population. However, whereas mortality has the potential to affect all sectors of a population, births are confined to those individuals who are fecund. Furthermore, human fertility, to a much greater extent than mortality, is directly influenced by the individual reproductive behavior, so that the parameters such as the age of first marriage and deliberate birth spacing have strong effect on the population fertility rate. From an updated sample of the Old and the New World Cemeterians in the Northern Hemisphere, a birth rate estimate is obtained ranging from 40.3 to 53.1 per thousand over 1000 years during the Neolithic fertility transition. Now let's summarize what we have learned so far in this module. Population growth after the development of agriculture has been one of the most dramatic features of human evolution. An estimated population was 10,000 years ago was 80 lakh individuals who inhabited the earth. By AD 1800, the world population reached 10 million souls. Male and female adult skeletal morphology varies across a continuum. Age and sex can be determined from the skeletal, which can be later helpful in assessing the number of skeletal, age at death, fertility and mortality ratios, and population growth patterns. Data from the ancient population, Europe and Asia shows that only a small proportion of the population lived beyond 50 years, and most died at about 60 or 70 years. In the case of the Middle East, that is southern Lebanon, the Herskovits and the Gopher studied the hundreds of skeletals that came from a hunter-gatherer series of the Neolithic period, that is 15,000 to 12,000 AP, and pre-ceramic sedentary groups from the Neolithic from about 12,000 to 8350 AP. For the former group, the estimated life expectancy at birth, assuming they are stationary population, is 24.6 years, while for the later group it is 25.4 years. Paleodemographers, however, have typically reported unexpectedly low infant mortality and adult age specific mortality that is abnormally high and that accelerates rapidly from age 15 to 50 years, resulting in few individuals surviving into the old age. The rise in the population density, domestication of the animals, and the increase in the work effort is the course of the Neolithic revolution, which increased the exposure and the vulnerability of the humans to environmental hazards, such as infectious disease, and led to the decline in the life expectancy during that period. The first period in the pre-modern times is Paleolithic period that lasts 3 to 5 million years. Subsequent period, however, are indicated according to the scale. Starting with the Neolithic period that concerned about 8000 BC and last until about 3000 BC. This was followed by the Bronze Age, which in turn were followed by the Middle Ages. Further, human fertility, that is to much greater extent than mortality, is directly influenced by the individual reproductive behavior, so that parameters such as age at first marriage and deliberate birth spacing have strong effect on the population fertility rates. From an updated sample of the Old and the New World cemeteries in the northern hemisphere, a birth rate estimate is obtained ranging from 40.3 to 53.1 per thousand over 1000 years. Thank you.